Now we're going to look at adaptive defenses, which is specific immunity, which is our third line of defense, which consists of humoral and cellular immunity. And the characteristics of adaptive immunity, it's specific. So we have specific cells or chemicals acting on specific antigens. It's systemic, so it doesn't happen in a specific location. It's in circulation throughout the body. It's not restricted to an initial site. It has memory, so the next time you're infected with the same antigen, you'll have a stronger and a faster attack. So it requires an initial exposure. With that initial exposure, priming takes place, which means each subsequent exposure, your body will be better prepared to deal with that specific microorganism. It involves two branches. The first branch of adaptive immunity is humoral, or antibody-mediated immunity. Antibodies are created, produced by B lymphocytes called plasma cells, which circulate in our body fluids. They'll bind to the target and mark them for destruction by either phagocytes or complement. We'll go into humoral immunity in detail in another video. There's cellular or cell-mediated immunity. It's our T lymphocytes that act against a target cell by either directly killing it or by indirectly disposing of it through chemicals that enhance inflammation or activating other lymphocytes or macrophages. And we'll deal with cellular immunity in another video as well. So before we get into these two types of adaptive defenses, we first need to look at what an antigen is and the cells involved in these two branches of the adaptive system. So an antigen is something that our defenses don't recognize as our own, but of something foreign. So they're going to generate an immune response. Most are large, complex, like proteins, but others are small and may just consist of a few molecules. Antigens could be complete or they may be incomplete. Complete antigens stimulate proliferation of specific lymphocytes. For example, when a lymphocyte, like a B cell, for example, comes in contact with a complete antigen, proliferation means they start dividing and become active. That property is called immunogenicity. Complete antigens also have the ability to react with the activated lymphocytes or the antibodies. That's called reactivity, and those are the two important properties of a complete antigen, which could be proteins, large sugars, fats, and nucleic acids. They're found on the surfaces of microorganisms, or something like pollen. An incomplete antigen, also called haptens, these are molecules that are too small to be immunogenic. They're not going to produce an immune response. So small things like peptides, nucleotides, hormones, but they can become immunogenic if they attach to our own proteins. These are our self proteins. So there's a combination of our protein and the hapten, the incomplete antigen, that can become immunogenic. Now this can cause a problem because now if it's immunogenic, our immune system is going to mount an attack on ourself because it's going to attack our self proteins that have attached to the hapten. An antigenic determinant is part of an antigen. So here's an antigen here, but the determinant is a specific area of the antigen that receptors bind to. That antibodies will bind to, or a lymphocyte will bind to. So any specific antigen can have multiple determinants. Here we can see antibody A binding to this determinant, antibody B to a different location, right, and so on. So this is a good thing because we can have different lymphocytes or antibodies working to destroy an antigen at the same time. Now, on the surface of our cells, there's a huge variety of proteins. And if they're in our own bodies, our immune system will recognize them as being self, meaning not foreign, and they're okay. So we call these MHC proteins after the major histocompatibility complex coded by our genes, because remember, our genes produce proteins, and they're displayed on the surface of our cells, which means our immune system will not attack them. They're unique to individuals. There's millions of combinations, but if we put a protein, an MHC protein, in an individual that does not create these proteins on their own, and that will be perceived as foreign in another person. Just think of blood typings that are not compatible. So our MHC proteins hold or display a piece of self-antigen or foreign antigen. If it's a self-antigen, one of our MHC proteins, then all is fine. If a foreign antigen is displayed on our MHC proteins, well then T lymphocytes can bind to them and act to destroy them. So our third line of defense, or our adaptive immune system, involves three crucial cell types. The ones we know already but haven't really covered in detail, but we will, are two types of lymphocytes, B and T lymphocytes, which correspond to humoral and cellular immunity respectively, and something we haven't yet seen, antigen-presenting cells, or APCs, that do not respond to specific antigens. So what we're going to do is go through these three types of cells. So let's start with our lymphocytes, our B and T lymphocytes, and go through how they develop, mature, and how they're activated. And actually we'll go through that in the next video.